So we have talked about feed additives for cattle and ruminants to reduce the belching of methane from them. I'm kind of adding this part on freshwater plants and algae, whether they can act as effective feed supplement to reduce methane emissions, because it just randomly came up here. The institute I'm at, Indian Institute of Technology, Bombay, sits on a lake, and the lake is completely covered by water hyacinths, and there is always a debate about who is releasing what and causing eutrophication and so on in the lake. And now a young new faculty, Jaideep, wants to uh, monitor methane emissions from cattle on campus. The campus is famous for its cattle population. They used to wander around freely, come into offices and classes and corridors, buildings, and so on. But now they have kind of started to herd them and put them into containment areas, separating the bulls from the cows, and so on. Can we somehow produce some algae in the lake and feed the algae to the cattle to see if we can reduce methane emissions? Why not, right? It could grow into something, because India has got a huge population of cattle, and we got rice paddies as well. So we want to probably look at the idea of reducing methane as an overall contribution to our nationally determined contributions as well. Okay, so what are the highlights of this paper? They do a very nice study of considering many algae, do some lab experiments in 2 steps, and then consider mixed algae, look at their DNAs and the emissions, and then show that there are some potential candidates. Current supplements to reduce livestock emissions cannot be produced at scale. So seaweed is one of the things that is considered, and where you have long coast it's possible, but then to move that seaweed from the coast to the interior may be quite a challenge. Australian freshwater plants and algae can inhibit methanogenesis in batch testing, so they have done some field studies. Algal mixes containing Spirogyra reduced methane by more than 10% and had high lipid content. So you also want to be sure that whatever you feed the cattle, the milk quality at least doesn't get worse. Maybe there are ways to increase their fat content and so on. And one plant, Montia australastica, reduced methane by 6.7% and contained flavonoids. We'll see what they are. These are potential alternatives to reduce livestock industry methane emissions. Of course, if you do a life cycle analysis kind of approach, then you have to see whether the efforts involved in making all these algal mixes to reduce emissions by 10% or 6.7% are worth the trouble. Nonetheless, it's a nice study anyways. So this is a graphical abstract. Can freshwater plants and algae act as an effective feed supplement to reduce methane emissions from ruminant livestock? Identification of candidate species is done. In vitro batch assay trials, so that's lab experiments with various candidate species and their mixes, and measure whether methane goes up or down or whether it remains the same. And then you identify DNAs for key candidates, so you can maybe mix and possibly exploit Spirogyra with some other addition. So Montia is one of the ones. And of course you have to track bioactive compounds, N3-PUFAs and flavonoids are tracked here. Methane, so let's just read a bit of the text. I'm not going to add much in terms of details, because I'm not an ecologist, and these are experiments, and if you're going to try it in your neighborhood, you will have to maybe find your own mix of algae or a specific algae that may work, which means you probably end up doing these experiments again. But this is useful enough to look at to see that people have already tried it, and there are potential candidates that can serve the purpose of reducing methane from cattle. Methane production by livestock is a substantial component of greenhouse gas emissions worldwide. The marine red algae, I'm not an ecologist, Aspergillus taxiformis has been identified as a possible supplement in livestock feed due to its potent inhibition of methane production, but currently is unable to be produced at scale. So whatever you identify, if you want to make enough impact and feed many cattle, millions maybe, then you have to produce that candidate at large scale. And then there is the issue of transportation, storage, etc. as well. Finding additional taxa that inhibit methane production is therefore desirable. Here we provide foundational evidence of methanogenesis inhibiting properties in Australian freshwater plants and algae, reviewing candidate species and testing species chemical composition and efficacy in vitro as well. Candidate plant species and naturally occurring algal mixes were collected and assessed for ability to reduce methane in batch testing and characterized for biochemical composition, lipids and fatty acids, minerals and DNA. We identified 3 algal mixes and 1 plant, that is Montia australasica, whatever its pronunciation is, with potential to reduce methane yield in vitro batch assay trials. All 3 algal mixes contain Spirogyra, although additional testing would be needed to confirm this alga was responsible for the observed activity. So you have to eliminate the placebo effect, I guess. For the 2 samples that underwent multiple dose testing, algal mix 1, predominantly Spirogyra maxima and Montia australasica, there seems to be an optimum dose, but sources, harvesting and storage conditions potentially determine their methanogenesis inhibiting activity. Based on their compositions, fatty acids are likely to be acting to reduce methane in algal mix 1, while M. australasica likely contains substantial amounts of flavonoids, apigenin and camphorol, which are associated with methane reduction. So you can identify pathways, and show causal links as well, which is important to show that what you are accomplishing is actually because of what you are doing. Based on their mineral composition, the samples tested would be for livestock consumption at an inclusion rate of 20%. Thus, we identified multiple Australian species that have the potential to be used as a feed supplement to reduce methane yield in livestock, which may be suitable for individual farmers to grow and feed, reducing complexities of supply associated with marine alternatives and suggesting avenues for investigation for similar species elsewhere. Right? So when you want to use seaweed and other marine species, then you are talking about coastal farms to, let's say, grow these, and then you have to move them into the interior for the cattle, whereas freshwater species may be easier to manage. Let's just look at some of the approaches. Freshwater plant species, algae and algal mixes collected for initial testing for indications of methanogenesis inhibiting properties and chemical composition. Samples were collected from the Barwon River catchment, so they are taking actual native species, and there are several other places like Callahan's Creek, supplemented from elsewhere from less common species. Sampling was undertaken in November 2021. Some samples were combined across locations if there was insufficient material from either location. Anyway, these are the plants, and then there are mixed species also, which I'm not going to read off. And here are the species names. Azolla, which you heard of, used I think to increase the fat content of the milk, which makes it more attractive for commercial purposes. And we mentioned already, sorry, not Montia, Monitia, or it's pronounced Monitia, australasica, and crispatum, and so on and so forth. And these are the locations and what they did in vitro assays: first round, second round, multi-dose round, analysis biochemical, proximate composition and fatty acids, DNA, and minerals. Okay, so it's a very nice detailed study. They do all these and produce these results. Summary of methane and gas production after 24-hour incubation. So they have first in vitro trial, second trial, and then the final results. Within the same column, treatments not connected by the same letter are significantly different from each other. So there is a lot of details involved that I'm obviously going to skip. Dry matter is incubated, samples with multiple members are also listed, and so on. Here is the samples, the species again, total gas yield, so that has to be measured, various nice units that I'm not going to get into. Methane concentrations in milliliters per 100 milliliter total gas, okay, and methane yield. So that's concentration, that's yield, that's in milliliter grams per some unit of methane, I guess. Okay, so no, DMI is dry matter incubated, sorry. Okay, reduction compared to control, the total gas, so negatives are good because that's reduction. Methane concentration and methane yield. So you can, you know, if you can measure all these things and argue for a robust case for growing these algae and feeding them, including them as feed additives, and can accomplish measurable significant amounts of methane reduction, that's good news. But again, in the end, you will need cost benefit analysis, life cycle analysis kind of things. Very brief, I'm going to just jump to some discussion and conclusion, and the discussion is much longer, but I'm going to keep it short. We identified 3 algal mixes and 1 plant. So here it says Montia, but in the table it said Monitia, so some error somewhere, that have potential to reduce methane production in vitro batch in vitro batch assay trials. All 3 of the algal mixes that were effective at reducing methane contained Spirogyra. Spirogyra was also found in one other sample that did not show anti-methanogens effects, but is nonetheless worthy of further investigation as a potential feed supplement based on these initial findings. No other obvious algal candidates were identified based on DNA analysis. Remember, if you are farming and adding supplements, then you would presumably reduce the volume of other fodder, so there may be co-benefits in terms of land use, etc., that we have to, and water we have to consider as well. Multiple dose testing indicated that algal mix 1, which included Spirogyra, reduced methane most effectively at a 20% dietary inclusion rate, when methane concentrations and yield were both reduced 21 to 24% range, albeit alongside a small but significant reduction in total gas as well at 3%. This contrasts with the first trial when algal mix 1 significantly reduced methane without reducing total gas. Other 2 algal mixes, you can follow up with the tables, significantly reduced methane yield 11 to 12%. However, total gas volume was also reduced and the concentration of methane was not significantly reduced. Okay, so there are details on why you have to worry about concentration and yield. These differences may be due to changes in parent material during storage or to interactions among the species present. For algal mix 1, a dose-response relationship with total gas was observed, with the highest dose appearing to affect overall fermentation rather than methane production specifically. This suggests that a 20% dietary inclusion rate is optimal dose out of the 5 tested for that mix, and there are many more details about measurements, biochemical measurements, that you can read up. Okay, jumping to conclusions, our study has identified several candidates among Australian freshwater algae and plants that can moderately reduce methane. In the 2 samples that underwent multi-dose testing, algal mix 1, predominantly Spirogyra maxima and Montia australasica, there seems to be an optimum dose, but sources, harvesting and storage conditions would potentially determine their efficacy. For algal mix 1, it's possible that combinations of fatty acids are acting to reduce methane yield. Montia australasica likely contains substantial amounts of flavonoids, epigenin and kaempferol, which have been associated with methane reductions in the past, and so represent a possible mechanism for the initial reductions detected. So you know the causal chain as well. Each of the samples tested would be safe for livestock consumption based on their mineral content at a dietary inclusion level of 20%. Combined, these results suggest further investigation of Australian plants and algae as possible feed supplements is warranted. Similar species elsewhere may also warrant additional investigation to identify a range of taxa able to act as possible feed supplements to reduce livestock methane emissions. Okay, just for fun, actually, I can show an image of the Powai Lake with its massive cover of water hyacinths. Let me pause and show you that just for fun. Okay, so I stole this image from Wikipedia. Thank you, Wikipedia, but every year I contribute. So here we go. This is looking from the side of the Indian Institute of Technology, and there is a lakeside road here which goes out into this region which is called the Boat Jetty, which is closed now, but there is the Hiranandani neighborhood on this side. There is a Westin hotel on this side, and lots of runoff coming into the lake, and IIT has, I think, reduced much of its waste being released, untreated waters being released into the lake, and the constant efforts that happen very frequently to reduce the massive amount of water hyacinths seem to fail, but efforts will keep continuing, and idea would be to see whether there are ways to reduce water hyacinths and also grow algae that can be used as feed supplements to feed the cattle on campus to start measuring methane, and then maybe get something for the larger scale out of it. Could be fun project, but